I want to talk to you guys about the assessment of vascular plant diversity in the HD Mountains, which is what my senior project consisted of. And I want to start out by acknowledging that the lands that we worked on and the lands that we collected on are on current ancestral Pueblo and Ute tribal lands. And I think that's important to acknowledge. So thank you. So just a quick little intro about myself. My name is Emma Cooley, obviously. Just recently graduated Fort Lewis College in December with an organismal and environmental biology degree. I got a minor in studio art. And I have a very strong love for scientific illustration, which is part of the reason I choose science and art together. So a couple examples of my work. I think that art and science meld really beautifully together. And I think that it's a great way to use all parts of our brain. And it's kind of what started me out, drawing me to botany in the first place, was I just loved spending time with plants and painting them. And I also, when I first transferred to Fort Lewis College, I started working in the herbarium with Dr. Ross McCauley. And that was kind of my introduction to botany as a whole. And I really loved working in the herbarium. And it was one of my favorite things that I had started out doing. And I really decided like botany was what I wanted to continue on and continue researching. And so that's kind of where my journey with this started. So I just want to start out by giving you guys a little bit of a backstory to what floristic surveys are. For those of us who don't know, some of us do. Flouristics are the study of local floras and documenting quantities, composition, identifying taxa. So what that essentially means is we're just trying to figure out what plants are in an area, who's here, who's living here, how many are there, what are their species, and where are they living, and who are they living with, and kind of just getting an idea of things like that. And it's a really good way to showcase the higher plant diversity of an area. And these are very large undertakings, like the one that we did in the HDs, which I'll explain about the HDs a little bit more. My project focused on a relatively small area, and it still was an incredibly time-consuming activity where we had to go out into the field and spend all day in the field collecting and things like that. And this kind of information that we get from floristic surveys can be really beneficial to prioritizing areas that may have really sensitive or endemic species that may need to be conserved or restored. And so it's a great tool for conservation and getting an idea of what's here and what should we protect and what's important. So just a couple of things like that. So to introduce you guys to the HD Mountains, if you guys are from around here, some of you may know about the HD Mountains, some of you may not. I still talk to people in Durango all the time who have no idea what the HD Mountains are because they're not as popular and as flashy as something like the La Platas with the big alpine. You can go skiing in the winter. They're just these kind of lower foothills that are not as well known. And they're also about 25,000 acres roadless, which means they're actually pretty hard to get to, which is another reason I think not as many people know of them, because there's less availability for recreating in these areas. And so most of this area has to be gotten there by foot. And so there's only a couple roads, and some of the roads go up through Ute land, so you can't actually access that easily. You need to get permits on Ute land, which is difficult. So the big main road is Spring Creek Road that you can go up, and it's a pretty rough road still, and it makes it difficult. So the HD Mountains are about 33 miles southeast of Durango, for those of you who know where Durango is. And the HDs, which is a weird name, was named for a 19th century cattle company, because if you search the HD Mountains on Google, you're just going to come up with some really high quality pictures of mountains, and it may be a little bit tricky to actually find what you're looking for. And so they span ownership from the U.S. Forest Service and Southern Ute tribes. They range in elevation from about 6,000 to 8,000 feet. And they're very important winter range for a lot of wildlife, including deer, elk, and mountain lion, because they're a little bit more mild in the winter and they're a little bit lower and are roadless and relatively pristine. So a lot of these wild animals really rely on this habitat for the winter time and to make it a little bit easier. So I kind of want to start and talk about what originally got us into this project and why the HDs, like why are the HDs important? How did we decide this was it? So The HD Mountains lie at the northernmost part of the San Juan Basin, which ranges from the southwestern part of Colorado to the north part of New Mexico. And it lies on a pretty vast oil field. And so there is already a lot of drilling that goes on in northern New Mexico and in Colorado. So there's definitely a lot of interest around the HDs as drilling because it's relatively untapped. And so in 1991, there was actually a really large proposal for adding about 115 oil wells around the area of the HDs. And eventually the local public kind of just decided that that was too much and they didn't want that. And so the proposal got shut down. There are still some oil and gas wells currently in the HDs. When you drive up the roads, you see the mules and things like that, but they are much less than what they could be. So talk about that. And I also would obviously like to bring up the San Juan Citizens Alliance because they're the original reason that we started this project. They were undertaking this thing called the BioBlitz of the HD Mountains, because the HD Mountains are also historically have had areas of interest of areas of high biodiversity and unique species we've seen from previous collectors as far back as the early 1900s. And so they started this BioBlitz to do this all-taxon survey with plants and mammals and birds and vertebrates and amphibians and things like that, everything that's up there. And our Fort Lewis survey only included the vascular plants, obviously, and was just the one growing season this past summer 2021 growing season. And what makes this area important, like I was saying with the oil and gas drilling and the San Juan Citizens Alliance acknowledged, is that there are actually a lot of unsold leases in the area for oil and gas drilling that have the potential to be sold for that purpose, which is part of the reason that they want to see what do we need to conserve in this area, what's here, and what should we be doing to acknowledge important wildlife in this area too. And there's potentially unique biota as we've seen. And so that's kind of the San Juan Citizen Alliance started it all, and well, thank them for that. So to start out with what my study outline was and kind of where we were, what we were doing, here's a little bit of a map on the left for you guys. So that red outline is the bounding box area of the HD Mountains, of the study area that we collected in and that the rest of the BioBlitz survey was being undertaken in. And they are a pretty understudied area, which is another reason that this area is of interest, is that, like I said, there's a huge roadless area, and so they're hard to get to. It's very remote, so we don't know what's in all the little canyons on the sides and things like that, and because they're hard to get to. And so we wanted to try and see if we could bridge that gap and maybe find some things that were in areas that were difficult to get to and make connections with past collections from past collectors that we've seen from early 1900s, as I was saying, that collected plants in this area and other animals and things like that. So I will go ahead and just talk a little bit about the past of the HDs, because there's been some pretty cool people that have come through. So the earliest recorded collections in the HD areas was from this guy named Charles Fuller Baker. He came through Durango in 1898, 1899, and did a lot of collecting around the Piedra River. And he was a pretty notable botanist of his time. He worked in Durango a little bit, and then he also lived in the Philippines and Hawaii and Singapore, and he was a teacher and did research out in those areas. And so he was a pretty cool guy of the time. And it's really, really cool to be able to see collections over 100 years old that are still intact today, like the Amelanchier on the right there. That's a herbarium specimen that he collected in 1899. And you can still see the green of the leaves and things like that. So it's just a really cool piece of history that we can have something like that in this area that we collected as well. There was also this woman named Hazel M Schmoll, who I think is just an amazing lady. She was the first woman to receive her doctorate's degree in botany from the University of Chicago, with work that she did in this area supporting her PhD. And she was the Colorado State botanist from 1919 to 1935. She mostly stayed near a Forest Service station on Yellow Jacket Pass is kind of where she did most of her collecting. And she didn't quite get out into the more western area of the HDs, which is where we did most of our collecting and things like that. But she was an amazing lady, one of like the first of her kind, just super cool. And there's a species of rare Astragalus schmolliae, which is named after her, which can be found in Mesa Verde. It's endemic to Mesa Verde. And so, you know, some of her specimens are about 100 years old as well. And there's one on the right there, the Wythia, is still green and yellow and holds its color. And it's just super cool to have that history again. And I think that I loved seeing her name pop up over and over again with all the collections that she made. It was kind of a cool thing to see. And a little bit more recently, this man named Ronald Hartman, who some of you may have heard of, is the curator of the Rocky Mountain Herbarium at the University of Wyoming. And him, along with Jeanette Flagg and Bernie E. Nelson, did a Flouristic survey on the eastern side of the HD area, kind of in the early 2000s, 2003, 2004. And he also was the founder of the Rocky Mountain Herbarium's Rocky Mountain Flouristics program. So he did a little bit more research in this area along with other people a little bit more recently. And he just recently died too in 2018. So over the summer of 2021, I'll give you guys kind of an idea of what we were doing. So in these two pictures, this is me and my academic advisor, Dr. Ross McCauley, who I mentioned earlier, collecting in the back of his truck. It's a really kind of fun activity, collecting all stuff. So kind of what we do is we drive around, you walk around and you look for flowering plants because the plants need to be flowering in order to identify them. It's a little bit tricky to identify things with just a leaf. So if someone comes up to me and they're like, what is this plant? And they hand me a leaf, it's almost nearly impossible unless I know exactly what the plant is, because we need to have the unique morphological structures found in the flowering structures and things like that, the reproductive material and things like that. So that's very important. It must be in a flowering state. And it can't negatively impact the population because we do dig these plants up fully and we shake the dirt off the roots. So we want to make sure there's not just this one super rare species that we're taking the whole plant of. It's kind of counteractive to what we're trying to do here. And we have to make sure that the specimens fit onto a standard herbarium paper size. So we have to bend them a little bit and kind of orient them. But you take some newspaper and you stick your specimen in the newspaper and you fold the newspaper and then you put some cardboard and then you put some blotter paper and you make this big sandwich, and then you wrench it all down and you press these big specimens. You can kind of see an example of that in the back of the truck there with Dr. McCauley. And so our collection dates were from April 25th to September 10th. And that's about the extent of the growing season at that elevation and in that area. And we went up about a total of 10 times, 10 different trips up to the HDs. And you can see on the right here, this map that I have, all of those red dots are collection events from myself and Dr. Ross McCauley throughout the period of April 25th to September 10th. And as you can see, so the red dots that kind of go up, that's Spring Creek Road that go up towards Pargan Mountain. And that's the main road. So it's definitely easiest to collect along there. And there's the Yellow Jacket Pass and a couple other areas that are easier to get to. We weren't able to get into those canyons quite as much as we had hoped, but hopefully in the future we can get to do something like that. So once we had these specimens all pressed, and we would press them, and then usually in order to dry them, there is a very nice fancy drying machine in the herbarium that we can use. But it's also July, and so we can just chuck them in the back of our car and leave them for a few days because it's so hot and it'll perfectly dry out the plants just as much as the professional one in the herbarium. So we would leave our plant presses and our plants to dry in the back of our cars for an extended period until we made sure that they were no longer had any moisture inside of them. And once we did that, we took them into the herbarium, and the real work really started to begin, I think at least. It was a very intensive work identifying these plants, because I had also not worked very much with a dichotomous key and floras and things like that. And learning botany is like learning a second language. There's so much terminology, I'm sure as you guys know, that is really tricky to understand when you first start out with this stuff. And so it was a learning curve, as Ross told me, as I got perpetually more and more frustrated. He was like, it will get easier. And I'm like, I don't think it will, Ross. And it did. It eventually did. And I finally was able to identify things a little bit quicker. And, so we used microscopes, and sometimes I had to rehydrate the plants with some soap and water and let it soak so I could open up these dried flowers a little bit better and look to the characteristics of them. So once they were identified, and hopefully identified them all correctly with Ross's help, and I used the Flora of Colorado, Sedges of Colorado, and Colorado Grasses. Those were my main 3 books that I spent a lot of time with and got very, very close with. And after all that identifying, the mounting and digitizing process began. And so the picture on the left is a picture of me mounting a specimen. And so what you do is you take your specimen and you put some pH neutral glue on the specimen, and then you put it on this cotton or barium paper, and you try and line it up and make it look nice. And I love this part because it really kind of added to my artistic side. And I thought every piece was just like making art. And I thought it was really fun. I really loved doing that. And then there's all these other, you have to put the locality information, the catalog number information on these specimens that you collect. And after those were all said and done, we got to digitize these. And digitizing is really cool because it means that we can upload it to the internet. We're taking these really high quality pictures of these specimens and uploading them so that they're accessible to the greater public and for viewing and for scientific purposes and further research, which is a really cool step. So in addition to the specimens that I collected and Ross collected over the summer, we also, like I said, from Hazel Schmoll and CF Baker, we incorporated other collectors' specimens that were recorded in the database SciNet, which is a database that is the Southwestern area database for plants. And so we had that bounding box, and anything that was in that bounding box from previous collectors were also incorporated into the overall species list that we had at the end. So I had collected about 286 or something specimens, and we added about 490 something, I think, specimens. Was it about 700 specimens actually, sorry, from the database. So there was a lot of collections that we had from Hartman and from Baker and from Schmoll and things like that. So we had a total of about 987 different specimens, not species, but specimens that was added to the entire species list in the HD area that we collected. So the one on the left there is from Hazel Schmoll, and then Baker and B.E. Nelson, who worked with Hartman. And it's just so cool to see that those specimens are still outliving us and will be for a long time. So the really good, fun stuff, I'm sure as you guys are waiting, what did we find? What was really cool? What was exciting? And there were a couple cool things that we found. I think if we had been able to get into those deeper canyons, there could be some really cool, interesting things down there. But what we did find was, from that entire compiled list of previous collectors and my collections, was 410 species were identified within 74 different plant families. Our most speciose families include Asteraceae, which had 85 different specimens, and asters, for those of you who don't know, is the family that contains sunflowers and daisies and things like that. Brassicaceae, 25 different species, and that is our mustard family. Fabaceae is 29 different species that we got, and that's our bean family. Poaceae, 39, was our grass family, and Rosaceae was 18, which is our rose family. And the picture on the right, our rubus, is a part of that rose family. The largest genus that we collected was Astragalus, and that's common name known as milk vetch. And our second largest genus was Artemisia, which is our sagebrushes usually. So if you're driving around New Mexico and all you see is flat, 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 and there's just a couple little things of green, that's usually going to be your sagebrushes, and that is the genus Artemisia. So I'll lead you guys through some interesting specific collections that we found. This guy on the left, Viola Natalii. is a relatively common viola in the area, but had never been seen in the HDs before. And so this was the first observed occurrence in the HD area that we collected. Just a cute little yellow viola. So it's not super uncommon, but in this area specifically, it's uncommon. And the one in the middle, one of my favorite collections, the Pterospora andromedea, is a really, really cool little plant. It's part of the Ericaceae family, which was the first record that we had of the Ericaceae family in the HDs. And for those of you who don't know, the Ericaceae family also gives us blueberries. And so this Pterospora, what's really cool about this plant is that it's achlorophyllous, meaning it doesn't have any green parts, doesn't have chlorophyll. As you can see, it actually has no green, which means that it relies on fungal relationships to get its nutrients. So they're usually found with Pinus species, because the Pinus species and this Pterospora, both they have fungal relations, which is a common relationship found in this area. And so that was one of my fun things to find, because it's kind of this cool alien looking plant. I was really excited when I found it. It's a relatively common plant, but I think it's still very cool. And then on the far right is our Portulaca. It was the first record of the Portulacaceae family. The Portulaca is a very common weed that you see everywhere you go, everywhere you walk around, in the gravel. It's everywhere. It's kind of just this small little weed guy, but it was the first record of it in the family, probably because no one thought it was relatively important enough to collect in the first place, but we did because that's what we were here to do. So we had collected over the survey 54 newly collected species in the area just from our survey, and the two families, Ericaceae and Portulacaceae, were the first of their families that we found. So this was one of my favorite finds as well, Fritillaria purpurea, excuse my Latin, it's always difficult to get right. And this was the first recorded collection within Archuleta County. And this is a locally pretty uncommon species that we see. And it's a really cute, really beautiful little lily guy that is just like, its common name is spotted fritillaria, because it has these little speckled purple and green and these really large stamen, yellow stamen. And so that was a really fun species to find. And we just, like anything, you're just walking around, you come on it, you're like, oh. And it was cool because there were a couple other populations around the area as well. So that was one of my favorite finds, little fritillaria there. Another interesting species that we found was Astragalus proximus. And this species is recognized by the Colorado Natural Heritage Program as apparently secure, which means it's kind of one step up from being secure and fine. But it's what makes it interesting is that it's a regional endemic to our area, southwestern Colorado and northern New Mexico and counties. So it's only found kind of in our area, not super uncommonly, but it's a very specific to this area. And it's considered imperiled and vulnerable in New Mexico, not as much here, but in New Mexico it is. And its common name is Aztec milk vetch. And it's part of our Astragalus genus that I talked about earlier in our family, Fabaceae, our bean family. And the last kind of cool guy we found was the Thelopodium wrightii. This was also the first recorded collection in Archuleta County. And it took a little bit of digging to find this because we couldn't find it in the flora of Colorado. It was found in the flora of the Four Corners region because it's found in New Mexico a little bit more commonly and not as much in this area. And it's kind of just tall Brassicaceae has these really long skinny fruits, and it doesn't look the most beautiful on paper, but it was kind of a cool plant to find up there. And as we're all very interested in the non-natives findings, so out of the 410 species that we found, 43 of them, about 10%, are non-native species in the area, 8 of which we newly collected in this 2021 study. And many of these species, as I'm sure you guys will all be shocked to find out, come along with oil rigs and being next to roads and areas of high disturbance. So places that are really gravelly, places that are walked on a lot because workers' shoes and vehicles bring in a lot of exotic seeds from a lot of different species. And so something like a cirsium, which is a lot of our thistles that we find, which we have a lot of issues with thistle. That was one of the non-natives that we found. Bromus tectorum is a non-native grass that we found. And So they were kind of sprinkled in areas, as you can kind of see in the background of those pictures. The sursum was in a ditch just off the road, and it's thriving happily. You know, I guess you got to give it sometimes to the non-natives. They're doing pretty well. They're doing their best. So that was kind of what we found as far as non-natives go up there. Nothing of particularly extreme high concern. And a lot of the non-native species, they are ranked by the Colorado Noxious Weed Act. And they are ranked according to how important they are to eradicate, to watch, and things like that. And it's dictated mostly by agriculture and how deadly it is essentially to cows, because if you have a plant that's invasive, that's really deadly to cows, that's going to be a level of high priority. So that's kind of how that's dictated in the Colorado Noxious Weed Act. And so I thought I would show you guys the final product of the fruits of our labor, this giant species list on the database on SciNet. So give me a second here and I will share my screen with you guys. So when you click that link that I had, it brings you to SciNet's database. And so SciNet's, this is the species list, this is my species list and the compiled species list of previous collections. And it has all of this listed by family, and it has all the different specimens, and it just goes on and on and on. As you can see, this was the biggest part of the work, was compiling and getting this list together and making sure that everything checked out and there wasn't something that was like, that's a Florida species, that shouldn't be here, and making sure everything was good and it was part of this list. And so I thought I'd just walk you guys kind of through the species list, because this is the interesting part, because it means that we can connect it with the greater population and you guys, and people can look on internet and see what was here. And so it makes it a lot more accessible to not just the people that made it. So we can look at things like the dynamic map. Load. And this shows the bounding box area of where we were and where we were collecting. And you can kind of zoom in on these collection events, and it shows you specifically what was collected here. So that specific one I clicked on is, like it said, Emma Cooley, or there's B.E. Nelson, and some of them are, you know, let's see here, we have to kind of mess with it sometimes. It's been a learning curve working with SciNet. But the cool thing about this is that you can kind of get to see who's here at a community level, like what's growing with this and who's growing with these, and what are all these collections and how are they growing together and how are they significant and how is that interesting. And so this is a really cool feature, these maps that just show all these collection events. And this is Spring Creek Road to give you kind of a reference of the previous map and how it looked earlier. So let's see. Thing. So coming back here, what you can also do is you can look for specific, you can look for a specific species if you're looking for one. So here I thought we would look for our beautiful Calochortus gunnisonii, and then you can click it and press enter, and it'll bring you up a specific detail of it. So it'll show you our family first, Liliaceae, and for those of us who don't know, the Calochortus gunnisonii is commonly referred to in this area as a sago lily. It's one of my most favorite plants, and I loved finding it up there. So if you first click on this link right here, it should take you to a kind of general description of what the species is and the collections and where it's been found and other digitized collections and other things that people have found it with and things like that. And so it's a really cool feature to kind of just get information of what is this species and who is it and what does it look like. And next, if you click on this link that says Emma Cooley 96 FLD, I'm not going to click it because I already clicked it earlier. It will bring you up to the specific collection that I made, and it will give you all this information about this specific collection that I made. It'll have the taxon, the family, the collector, the date it was collected, additional collectors, Stephanie Matlock and Doug Craig. That is my mom and my stepdad, who were very gracious to come collect with me. And it gives you the locality, United States, and it tells you kind of where it is, just off Spring Creek Road. I could have been a little more specific, but when you get really specific, it's so that if someone was like, I want to go find that plant. I want to go find it today, and I'm going to go do it. They could, because there's the information of where the species was found. And it can get really specific because we also include the coordinates on it and things like that. We also have elevation, the habitat, kind of where it is, you know, what kind of soil was it in, and the associated taxa. So we have Quercus artemisia, Pinus, and Juniperus, which gives us an idea that it's a sagebrush flat with pinyon juniper forest and some Gambel's oak as well. And so you can click here and click open large image, and it will actually bring you to a really nice picture of the specimen. And so you can zoom in, because these are high quality pictures. You can zoom in and see very interesting little details of these plants. And I think this is one of my favorite features, because you can just like really get in there and see all the really cool different parts of the plant. And so I think that's definitely one of my favorite uses of this. So you can also, when we come back here to this the SignNet area, you can click on simple map, and it will show you where exactly this specimen was found based on the coordinates. And so it shows you right there, that's where it was found. So you can go find it. If you too want to go see the super rare sago lily, you can go find it as well. It's not that rare, but it's beautiful. So I'll just go ahead and bring us back to our PowerPoint here. So just wanted to share a couple of fun things before I end this up. We got very stuck in the mountains. This is my very wonderful partner here who took us up in his truck, and we got incredibly stuck. But we had some very wonderful life lessons of getting a truck out of the dirt and out of the woods. And eventually we were very kindly rescued by a friend who came and drove all the way up there and pulled us out. So thank you for that. And obviously we made mostly plants are mostly friends. As you guys all know, to a botanist, plants are our friends, and they're our closest friends, and not always only our friends, but you know, we also make friends with cute little horny toads and some cows up there. And so it's kind of fun to get some animal time as well as plant time up there, which was great. And finally, I just want to acknowledge everyone that helped me with this. I want to thank Fort Lewis College and Desert Ecosystem Analysis for grant funding and support, and to the San Juan Citizens Alliance for starting this whole thing and putting together the Bio Blitz, the San Juan National Forest for allowing collections. And I also want to thank Stephanie Matlock, Simon Mount, Doug Craig, Lydia Thompson for going out in the field and bearing the botany world and collecting with me and running around doing weird plant stuff. And I also want to thank Savannah Bell and Albany Dubacher for their help in the herbarium. And lastly, I would like to extend a huge thank you to my professor, Dr. Ross McCauley, who has helped me throughout the years and spent hours and hours working with me and identifying and helping me and just kind of showing me how the whole botany world is and introducing me to it. So thank you. And now we can open this up for questions, and I can be happy to chat with you guys a little bit about my project and hear what you guys had to say.